and welcome to the latest A Spotlight On interview. Today, we're going to be talking about genetics and drug discovery. I'm joined by Erik Ingelsson. Erik, could you please introduce yourself and tell everyone a little bit about what you do?
Sure. Thanks, Bobby, and also thanks for inviting me to this interview. So I'm Erik Ingelsson, and I'm Senior Vice President of Genomic Sciences at GSK. Before joining GSK, which was about 2.5 years ago, I spent about 20 years in academia, both in Europe and US, and last position as a professor of medicine at Stanford University. So at GSK, I lead a department of about 300 scientists, and we are working on a range of different methods in genomics, ranging from human genetics, bioinformatics, computational biology, genome biology, including both CRISPR and other gene editing tools and iPSC models, to omics, both single cell and bulk type of sequencing approaches, mass spec approaches. So really broad set of applications within genomics. We're working both everything from early target identification and validation all the way through the whole pipeline.
Thank you. And to dive straight in, what are some of the current challenges in drug discovery and development? What are our success rates in bringing something to patients?
Yeah, so the main challenge for drug discovery and development is really the low success rate. So we're looking at new molecules entering clinical trials. We're about 10% success rate, meaning that about 10% of medicines that we start with a phase 1 trial ever reaches the market, ever gets approved and gets to patients. So that's a really low success rate, really. And if you go earlier, obviously it's much lower than that. So if you look at new programs that we start where we have a target and try to find a medicine, it's less than 1% success rate. So obviously that's really the biggest challenge in drug discovery and drug development. There are obviously additional challenges, but I would argue that that's the main thing. And thinking about why that is, I think we've been relying a little bit too heavily on either correlations, so looking at things like biomarkers of gene expression levels in patients versus non-patients, which is a correlation but not necessarily a causation. And we have also relied quite heavily on in vivo models, a lot of mouse models, for example, in drug discovery. So I think that those are some of the key challenges that we've been facing.
Thank you. And what are some of the current timelines and costs to bring a drug to patients?
Yeah, so the timelines are typically, up towards 15 years. And it obviously will depend a lot on the type of drug, the disease. Certain diseases, the randomized clinical trials will be very large and long, whereas some other, especially rare diseases, but especially orphan drugs, etc., you can kind of have a different path, and some oncology also different path. But so it will vary a little bit, and it also depends on where you start. If you have more knowledge on the biology, you might even have a medicine already developed for some other indication. Then it could be faster. But typically, I would say at least 10 to 15 years, and sometimes longer. In terms of costs, there are various numbers out there, but generally they're high. It's recent numbers I've seen from scientific publications are in the range of $1 billion to $3 billion per drug that reaches market, and that's when you're taking expenditures for all the failures into account as well. And that's clearly an unsustainable situation. And it's driven by the things we talked about. It's driven by the long process, and it's driven by a lot of the failures. And especially since, many of the failures happen really when you get into phase 2 and phase 3. And these are the most expensive parts of the drug development process. So that's also what's driving the costs a lot.
Thank you so much. It's always fascinating to know the pipeline as well. So you talked about mouse models and the challenges they're presenting as well. So looking at our historical approaches, what have they been, and what are the growing and emerging options?
Yeah, so in terms of historical approaches, For target-based discovery, so that means that we start with a specific target and try to find, medicines for it, molecules. Then we have started a lot from known biology. So known pathways, known biology that has already been disentangled in great detail, quite anchored on animal studies, as I referred to, focus on target classes that we know that we can find molecules for, so such as kinases or GPCRs, so certain drug classes. So that's one approach. Another approach that's been used a lot is phenotypic screens. And that's basically an approach where you do a molecule screen trying to find new molecular entities with the ability to alter a cell phenotype. So you're not really looking for a specific target, you're looking more for a change in the phenotype. And you typically would use cell-based or animal models for that. And those have been kind of the traditional ways of working. Now, a lot of the pharmaceutical companies that now are pivoting more towards a stronger focus on human genetics, computational biology, functional genomics, and artificial intelligence, machine learning, AML for short, as the starting point. And the idea behind this is a couple of things. So one thing is that we're trying to expand into biology that haven't been looked at before. So without the constraints of what's already known, so more hypothesis generating or hypothesis free approaches, which was really the big change in genetics, 15 years ago when genome-wide association studies came. But also to more strongly focus on humans as the model system for target identification and validation. So rather than doubling down on mouse studies, we and a lot of other companies are focusing much more strongly on humans, meaning a lot of human genetics as a starting point, and then working with different type of in vitro models based on human cells for some of the target validation work.
When it comes to identifying a drug candidate, there's obviously a myriad of factors going on. It's very complicated. If a dream candidate landed on your desk, what makes an ideal candidate?
Yeah, so it's a couple of different factors. So I think probably the most important thing is that it addresses an unmet need, meaning it is actually for a disease where we don't have good medicines. We have patients in need, and they're not being treated optimally. That's the main driving factor, I would say. But then there are obviously a lot of different ways of trying to get to that. And I think in terms of those factors, having a strong biological connection, primarily from human systems, so that's where genetics is really helpful, comes in. So very strong reason to believe that this target is involved in either the development or the progression of the disease, pointers towards causality, where genetics helps. So that's kind of another second important factor. A third important factor is that we have a feasible path to a medicine. And what I mean by that is that we think that this protein can be drugged. It's like we can find, it's tractable. We can find some type of way of modulating it. And also that we have a translational path. So we can kind of, when we have a molecule, that we have some type of biomarkers for target engagement, for pharmacological effect. So we actually can measure that the molecule is having an effect. And then the other things would be, lack of safety signals would be one thing. And so meaning that we have no indications that it would be dangerous to treat with this, obviously, in our preclinical systems. And then obviously also that there is some novelty there, so for us as a company to develop something, it would be some new type of medicine hitting a new type of pathway. Otherwise it wouldn't, and that ties into my very first criterion, otherwise it wouldn't really benefit patients. So.
Absolutely. And you've talked there about some of the benefits. So let's dive into that. What are some of the key benefits of a genetics first approach?
Yeah, so working with genetics, we can get indications of causality in disease development and or progression. It's a human system, so that gets around the problems with correlation and causation of correlation in biomarker studies, and also gets away from mouse studies. So if you think about this schematically, maybe we can pull up a figure here and kind of side by side look at the randomized clinical trial, which is the ultimate trial we need to do before registering a drug, and then compare that with the genetics study. They have a lot of similarities. You're starting with your eligible population, and then in the randomized clinical trial, you're randomizing individuals into either getting a drug that lowers, for example, blood pressure, or placebo, and then you preferably do it blindedly. And then you follow, in this case, blood pressure change over time and also cardiovascular disease. Now, with the genetic study, you have a similar situation. So you have the randomization that happens at the conception, and then you're comparing the blood pressure lowering variant with the neutral variant, so which is the placebo. And then you follow blood pressure changes and cardiovascular disease over time. So it's a conceptually attractive, and that's why some of these types of genetic studies are called Mendelian randomizations. So Mendelian, happening at conception, and the randomization of the alleles. So that's kind of the basics of why human genetics can model and predict causality in a way that other methods really can't.
Fantastic. And you've talked about the genetic evidence, you've talked about the biomarkers and how that's helping. So what other genetic evidence do we need to consider in the context of drug discovery? Can you define that for us?
Yeah, so what we really mean with genetic evidence is that we have evidence of a causal gene. So when we start with genetics, we typically have a statistical association between a region in the genome and a disease, or a disease-relevant phenotype. It could also be blood pressure or LDL or some other type of biomarker that is relevant for the disease in question. We typically mean that it's significant, taking all of the multiple testing into account. So we say genome-wide significant, it's replicable and robust, but that's not enough. That only points us to that region, as something in that region is telling us that there is a causal association between something in the region and the disease. Now, we need to connect the dots, and the first step there is really to connect the dot between the genetic variation and a gene, one gene. And that's what we call variant to gene studies, V2G. So when we talk about what genetic evidence is, in reality, we're really talking about the strength of the evidence of a causal gene in the region and knowledge about which gene that is, because that's where we need to start our drug discovery efforts. We need to understand, a specific target. We're typically not, it's not enough for us to know that there is something in the region. We need to know exactly what it is in the region. So that's typically kind of the starting point. And then additionally, it's beneficial for us if we know about the directionality. So is this gene upregulation or downregulation going to be what we're looking for? So we're looking for an agonist or an antagonist. That's basically also something very important for us in the early steps. So I would say that a lot of the main difficulties right now in translating human genetics into drugs are related to this, like variant to gene mapping. Say from the big number of associations that we're aware of, I would say that maybe up to 20%, 15-20%, we can be very confident about the causal gene. It's a coding variant or very strong co-localization or other approaches that can, we're very confident about causal gene. In the rest, like 75 to 80%, there are either no evidence for a causal gene in the region, or it points to several and we're not really sure. So that's kind of a big challenge right now in the field. Sequencing can help. So we're working a lot with both exome and whole genome sequencing, but it's not the, solution that will solve everything suddenly, because a lot of the variation is actually kind of a lot of the signals are really going to more regulatory elements in the genome. So we'll kind of need to work with other things as well. The other big obstacle typically would be the directionality. So again, before we can start the drug development program, we need to know if it's if we're looking for an antagonist or an agonist. And even with sequencing, quite often it won't help. We can have some evidence or some strong beliefs that this is a loss of function variant, and that's good. But quite often, most of the time, it's a missense variant, and we don't really know the directionality. So we'll have to work with functional experiments to figure that out. So those are probably the main things in terms of challenges that are related to genetics. And then there are obviously a lot of other challenges in the early stages that are more related to kind of the first experimental approaches, which are, do we have the right model systems? Do we have a good in vitro model that reflects disease? A lot of these hits that we're finding for genetics as well are very novel in terms of biology, totally unknown pathways and unprecedented proteins and pathways. So that is tricky. So those are some of the challenges we're working through as well.
Some big challenges there as well. So another challenge is that historically, clinical trials and our approaches have often lacked diversity, especially in regards to sex and ethnicity, which impacts patient outcomes. So what do you think we need to do to ensure our future endeavours are representative and unbiased?
Yeah, so this is a very important issue for GSK generally, and also for my department. And we have a very ambitious DEI, so diversity, equity, and inclusion agenda that centers around different aspects, both related to people. So that would be things like recruitment and development of diverse talent and having good representation there, but also business. So that would be working for health equity by inclusive research, access to healthcare, clinical trials that reflect different populations and minorities, but also communities. So that's about expanding scientific opportunities in underrepresented communities to level the play fields. So we have a very broad agenda, and it's been moving beyond. We've been working for several years already a lot with the people part of that and diversity and inclusion in terms of how we recruit and develop, but we're now expanding much more into kind of the business and community aspects of DEI. So in Genomics specifically, we're working with a whole range of projects, actually hitting all of those different kind of pillars. Again, ranging from trying to get a more diverse workforce. We're working very hard with that, and it's a very important thing for me, but also to several collaborations and interactions with scientific organizations, as well as educational organizations such as historically Black medical colleges, and so to also kind of broaden the genetics, both to talent, but also to education earlier on, and also to new large research studies in underrepresented minorities. And there are several pretty big efforts ongoing right now that I hope that we can be able to launch within the next year.
That's fabulous. Thank you so much for sharing. And you've talked there about the novel identification. So it sounds like sometimes it's luck. So how can genetics drive the therapeutic area unconstrained approach? How opportunistic is your work?
Yeah, so GSK has taken a pretty deliberate position here where we are unconstrained with regards to disease areas. So we've been really trying to go for the biggest opportunities for developing medicines that can make a difference for patients in need, as I talked about before. And genetics has played a really important role here because we've let genetics drive the approach without restricting ourselves to specific therapeutic areas. GSK is not like an immunology or respiratory company only, and meaning that we're not only looking for new targets in those areas, but we actually go wherever there is a good opportunity in terms of finding targets using genetics. So that means that we've been pretty unconstrained and also opportunistic in the way we work. Now, over time, we've been working with this strategy for about 4 years. now after Hal Barron took over as the president in 2018. So over time, there has been a development of stronger emphasis in some therapeutic areas that has been driven by the genetics, really, to a large extent. So now we're starting to see clusters of some diseases where we have a lot of like early targets and also later programs. But we're also proceeding with programs that are totally opportunistic, totally kind of a disease that we wouldn't have done, if we would have said, let's start with this disease. So I think we're still pretty opportunistic in terms of that.
And that sounds so exciting. If we look to the future, what are you excited for in the next 5 to 10 years? What are your hopes for the future?
Yeah, so I think the integration of human genetics and other approaches in genomics, so computational biology, functional genomics approaches, gene editing and sequencing, and also with machine learning approaches, I think the integration of all of that will really mean a step change in how we think about this. We have changed how we do this at GSK over the last 1 to 2 years now in terms of that link. I think it will lead to pretty dramatic steps in terms of how we link the genetic associations to causal targets and understanding of the biology. So we're doing a lot of very exciting work in that area, really doing things at an unprecedented scale in terms of how we work both with the human genetics and the functional genomics, gene perturbations and things like that. So I think that's kind of an area I'm really excited about. I'm also excited about having access and getting access to new data sets, both more diversity, as we talked about already in terms of ethnic background, but also data sets with deeper phenotyping, being able to look at progression of phenotypes and subsets of disease, etc. So that's something I'm very excited about. And then also new modalities that can open up more of the kind of undruggable genome right now. I think that's also kind of a third thing that is super exciting. Also for me personally, I'm also excited about expanding what we do with genomics beyond just the target identification and validation, and having a kind of more impact on the whole pipeline, which we are really expanding quite dramatically right now in our work.
Lots to look forward to. Thank you so much for sharing. I've learned so much today. I'm excited to see what the future of drug discovery looks like and the impact of genomics. So thank you for joining me.
Thanks so much for inviting me. It was a great discussion.
Thank you.